So as it was mentioned, I will present you some work that aim to combine different approaches to determine or to characterize the diversity of biofouling. And so this work was performed mainly by Aurélie Portas, which was a postdoctoral researcher in the project, which was supervised both by FEM and University of Toulon online. And this work was also performed in collaboration with the people from the University of Bretagne Occidentale for the especially for the morphotaxonomic identification. So yes. So traditionally, studies of biofouling community, and as Norwen has just presented, was mainly based on morphotaxonomic approaches. So these approaches are very interesting because they can give quantitative data, so they can give the real abundance of species. They can give biomass of the biofouling, and also some information on the size of the organisms. But this approach is also time-consuming, require strong taxonomic expertise, which is quite rare now, and this approach also tends to underestimate discrete, cryptic, or small species. So the idea was to look at other approaches, and you probably heard about the eDNA approaches, so the environmental DNA approaches that emerge from some years. And these molecular methods have the capability to detect a much wider range of taxa in general, including rare species, early stages of invertebrates, microbes, especially micro-eukaryotes, but also non-indigenous species. Of course, we have to be aware that this eDNA approach is not a perfect method, we can say. There is also some limitation, especially because there is only a relative abundance data, and in some cases only binary, so presence absence of the species. The data will greatly depend on the selectivity of the primers, so which are how we will make molecular analysis without going into details. And another point which is really important for this approach is the database accessibility and completeness. So this point makes that we finally have two approaches with, of course, pros and cons, and we decided to try to combine this two approach to have a better view of the biofouling community. So the molecular approach was developed in my lab in the University of Toulon. The validation of the DNA extraction was already performed during one of the previous projects that Lénaïque mentioned. So it was BIODHYL, and it was during the PhD of Aurélie, because Aurélie is here with us since for a long time. And can say, and the validation of the genetic markers and the associated primers and database was also performed during this PhD and during the BIODHYL project. And so during the BIODHYL project, we wanted to try to find a way to combine these two sets of data, which have different characteristics, and it's not so tricky to combine them. So the data set was based on ABIOD+, so you see the different sites we already talked about. So 2 in the Atlantic Ocean and 2 in the Mediterranean Sea. So these are the same sites that were studied in BIODHYL with different time of immersion. And we will work with these samples with the molecular data and the taxonomic identification. So for the DNA, just to remind, we have to crush the biofiling to extract the DNA, to amplify it. So specific region of the DNA with different markers, because we will see that they don't have the same results in terms of coverage. And then have a sequencing, huge sequencing, because as you can see, we will get at least 60,000 reads per samples. Then we have the bioinformatics step, and the use of this database I already talked about. And then we get a richness and diversity profile with the composition taxa, which is something similar that we get with the morphotaxonomic determination. So as I already said, that was performed by colleagues of the UBU. And so the idea was to combine these two data sets with the same type of data, so richness, diversity, but which could be different. So the workflow was this one. As you can see in the, perhaps I can have a thing. Yes. So here you have the DNA data with the two types of gene, and you can see, for example, that we don't have the same number of species with these two genes, and you will see after that, in addition, that will be not the same species. So we combine these two set of data, and you have an harmonization. I don't want to go into details. We can talk about that if you want, but the database are not with the same number of taxa. And the morphological approach, so you can see that there is also different number of species will then be integrated, and we will have a combined data with the redistribution of abundance. This is a very tricky step because, as I mentioned, we will have full abundance with the morphological approach, but we will have in some case only presence absence for the molecular data. And then the last step was to verify that the taxonomic affiliation we get are, can say, coherent with the species or the samples, sorry, that we get. I don't want to go into details once more, but we have some pattern like that to decide which could be the best way to retain an affiliation, a taxonomic affiliation, depending on some database and some also presence or not of the invasive species. And I will be back to this issue. So first results of this data. And you can see clearly here, you have the three types of approaches with the different species. And you can see that this is the data from the 18S DNA analysis that give the greater number of species. But you also see that there is a very few number of species which are shared by the three different sets of data. So finally, each method detects different species, and that is what is interesting, you can say. So if we look at this number of species and we combine them with the process, we will have a much more higher number of species than for each of the different one individually. Then going to the diversity of these samples. So I will show you some multivariate analysis. So it is quite the same type of analysis that show Nolwenn in the previous presentation. So to just to understand how to read these figures, each point is a sample. So it's a community, a biofouling community, and the more the points are close, the more the community are similar. And of course, it's the opposite. If they are very different, they will be far one from the other. So when we look at this, which is based only on the morphology approach, you can clearly see that there was a difference depending on the geographic pattern. So the ocean, you have the Mediterranean samples here and the Atlantic here. And what is interesting is that if you look at the different set of data with the two marker, the molecular marker, you have quite the same information for these geographic differences. But if you look at the other point, and the other point will be, of course, the length of immersion, so the time the sample will be in water, there is very different patterns, and there is not something very clear, perhaps except for this one, which is quite clear difference between the 6 and 12 months. And we make a correlation between these patterns with the Mantel test, and you can see that the values are not very good. And that means that there is not exactly the same repartition of the community. And when we use the combined data set, we have this pattern, and you can see that we keep the clear separation between the ocean, the Atlantic Ocean and the Mediterranean part, but we also seems to have more clear separation between the duration of immersion. So we compare the spatial dissimilarity and perhaps improve the temporal clustering. When we look at the composition now, this is a mean. You have the mean composition for the three types of approaches. And so you can see clearly that there is huge differences. And if we look at the morphotaxonomic analysis, this is the one with the lower richness, so the lower number of species. And mainly there are some Jassa, Caprella, and Mytilus, so multicellular organisms. And considering the molecular approaches, so there is clearly higher diversity, and especially for the 18S rDNA, you can see that there is, interestingly, in this part, half of the diversity is represented by microeukaryotes, so microbes, not bacteria, microeukaryotes with only one cells. And these organisms are less studied and less known, and it could be very interesting to have much more information on that. And we can see also that with molecular data, we will have information on algae, especially red algae. And for the COI, we come back to organisms more close than with the morphotaxonomic, with pluricellular organisms, bivalvia, hydrozoa, and Malacostra. So when we combine the data set, we get these results, and you can see that this is we have clear differences between. We keep these clear differences between the different sites. but we also see that there is a higher diversity than just with the morphotaxonomic. And so we can imagine some more specific characterization. Another point which is interesting is the non-indigenous species. And so we make a specific point on that. And you can see that with the morphotaxonomy, we only identify 4 species. With the 18S eRDNA, we identify 9 species, and for the COE, only 2. So once more, the molecular approach will give more a higher number of species. And I can see that there is only one that is common to all of this, which is the Methylucellulus, which is a non-indigenous species from Mediterranean Sea. Then we talk about the environmental drivers. And so I don't want to develop, but for this data, we use the data from the Copernicus database. And the aim was to try to correlate this community that has already shown you with the environmental parameters. And these parameters will be represented by vectors. And you can see clearly here that we have some set of parameters that are associated with the data in the Mediterranean Sea, especially with higher salinity, temperature, but also current. And another set of data of parameter, which is associated with the Atlantic, especially the nutrients and the wave, which is very interesting because that means that these hydrodynamical parameters are very important for the biofouling in offshore sites. And this was confirmed by the use of the variance partitioning, which is a way to quantify the involvement of the parameter in this community structure. And we can see that the most important parameter are the physico-chemical parameter, because it's the highest number, but hydrodynamics here, the second one. So that means that this hydrodynamics parameter are really important in terms of the structure of the community of biofouling in the offshore sites. So take-home messages: the combination of morphotaxonomic and molecular methods enables an enhanced detection of biofouling community in offshore environment. Each method has its own advantages. Morphotaxonomy provides quantitative data, visual confirmation of especially dominant macroorganisms, but the metabarcoding, so the molecular approach, reveals the broader spectrum of biodiversity, including microkaryotes and cryptic or early stage taxa. This integrative combination of methods, including the geographic filtering, allow to correct the methodological basis and significantly improve, we see, the taxonomic resolution and species detection. And the combination of these methods also improve the detection of non-indigenous species, which is, of course, something really relevant in this environment. So now, considering BIODHYL, we just have to apply this approach to the data that Nolwenn talked about, depending on the depth. And so this is an ongoing work. So thank you for your attention.